In this video, I'm going to be talking about identifying cancer cells using H-ferritin nanoparticles. H-ferritin nanoparticles are a reagent that can be added to cells that are suspected of being cancerous, and it can be a really small amount of cells. So if it was going to be used in a hospital to diagnose cancer, you would need only a really small piece, so you wouldn't need to have a huge biopsy. And they can also, these particles can detect cancerous cells at a very low concentration, so before a tumor has actually formed. So that would allow a doctor to recognize a cancer before it has a tumor formed and before it's reached the blood supply, so it won't spread as further is the idea. And so basically how the idea is that these nanoparticles are a protein that's actually produced by humans, and scientists load it with iron oxide, and then the iron oxide inside is attracted to human cells, that the receptors on human cells that are responsible for collecting iron for the cell, because the cell uses iron for other chemical reactions. And but most importantly is that some cancerous cells, not all of them, will overexpress this kind of receptor. So off the top of my head, there's two would be certain types of breast cancer and certain types of colon cancer. They'll overexpress these. And so the idea is that these nanoparticles will bind to the cancer cells, which have a lot of these receptors, and they'll bind to them a lot more than they would to healthy cells. And then a scientist would actually, you can't see the nanoparticles, they're basically invisible. So you have to stain for the iron that's actually inside here of the protein shell. You have to stain for the iron, and that's how you tell where the nanoparticles really are aggregating at. And so in the middle here, there's kind of a diagram of what the interaction of the proteins are. So the TFR1 receptor is the receptor that the nanoparticles bind to, and it's in charge of collecting iron. And again, it's overexpressed in certain cancer cells, but it has a really high affinity for iron. So kind of like an electron affinity is something with a high electron affinity will release energy when it gives off an electron, or when it takes in an electron. And it's kind of a similar idea here, where the receptor really wants to bind with the iron inside of the protein shell, and so it binds to it, and that's a lower energy state. And so if you have a lot of these receptors, then all of the nanoparticles that you put in the solution are going to bind to those receptors. And then which you can see here, this is an example of that, and you can see the cells, and the nanoparticles are actually highlighted in blue because the iron has been stained blue with this Prussian blue stain. But this is actually what a pathologist would see when they were identifying a cancerous cell. And so basically, it's pretty simple to do in theory. It's so you take these colorful circular human protein shell, load it with iron oxide, and you would soak your cell tissue sample that you think might have cancer. You would soak it in these nanoparticles, and then you would take it and put it on a slide and stain it with the Prussian blue. And then you would, ideally, you'd be able to compare between known sets of samples of these tissue stains that, and so you could tell if it was cancerous or healthy. And so, I'm gonna move my picture over here. So also it's really an important innovation because it would allow a doctor or a pathologist to detect a cancer or cancerous cells at a very low concentration. And that's going to improve survival rates of people who have cancer, because cancer is really hard to treat once it reaches the blood supply. And so if we could detect it with these nanoparticles at a really low concentration, that would help people to not die from cancer. But kind of as I said earlier, it's very you can't do these experiments on healthy tissues in the lab because healthy cells in the lab will just die, which is like, if you've heard of HeLa cells, then that's kind of where we started hearing about that, where cancerous cells can kind of be immortal. And that's why scientists do experiments on them. But because of that, there's not really any information about how these particles bind to healthy tissue and healthy cells that express the TFR1 receptor. And so if someone were to stain a biopsy sample with these particles and do this procedure, they wouldn't really know if it was cancer or not. It would probably light up some blue, but they've only really been doing the experiments on cancerous cells. So there really needs to be more research involved with healthy tissue, but that's very difficult to do because, like I said, those cells tend to die in the lab very quickly. So yeah, but this whole field of using nanoparticles to stain tissues will, in the end, hopefully be able to detect cancer much earlier, and hopefully that will be able to increase the survival rates of people who have cancer.